Hello, botanists. This is Courtney, and I'm here for our very first lecture that you're going to find on our Class Jupyter site. Now, really quick, since this is our first time, a couple things that you need to make sure you have. One, do you have your lab notebook? You're going to be writing down anything that I write down. You're going to be drawing anything that I draw. So the other thing you need to get in addition to your lab notebook is you need to get some pens, pencils, and colored pencils. I use color a lot when I draw or diagram or model organisms because it can be really helpful to understanding. So grab some colors. You need to pause me, and come right back. All right, so in these lectures, we're going to be shifting between, well, me talking, showing you pictures, and diagramming things. And so let me just start sharing my screen. To share this. So I'm going to be posting the PowerPoint slides on here as well. But I'm going to be broadcasting or showing you some of these here as well. So today we've got two basic things that we're going to be doing. We're going to be looking at the basic plant cell, and then we're going to be looking at something called plastids. And it's totally okay if you've never heard of these things. That's why we're here, right? We're here to get exposed to this information so that when you come and meet me in lab next week, we can just jump into it and get to work. So as far as how this course is structured, we're starting the course at the cellular level. And so we're going to start by building a plant cell. And so today we're going to be talking about those things that make plant cells unique. After we've covered cells, we're going to take those cells, put them into tissues, then put those tissues into organs. And if we're talking about plants, the organs aren't stomach and skin and brain and things like that. They're leaves, stems, and roots. So things that you are already familiar with just from eating and interacting with the world. Now we're going to start with plant cells because that's how I structured this course. But the plant cell is really going to be our model for the cells that we're going to see in all kinds of other creatures. So we're animals. We're not really going to focus too much on animals, but we're going to spend a lot of time looking at plants. We're going to spend some time looking at fungi, and we're going to spend some time looking at protists. And protists are things like algae and diatoms and slime molds, and some really, really diverse creatures. So I want to shift this and shift to this piece of paper. So this is where I'm going to start drawing, and you're going to start taking notes in your notebook. So we're starting with a cellular level. And cells, remember, are the basic structural and functional units of living organisms. So basic structural and functional units of living organisms. Well, that sounds kind of obtuse. What this means is that life is made of cells, right? That's what they're made out of. And everything that an organism does at the cellular level, it does at the multicellular level. Some organisms are going to be made up of just one cell, like some algae and amoebas and diatoms and tiny little creatures, whereas others are going to be made out of trillions, right? Trillions and billions of cells working together to do all the things that a multicellular creature does. Now, all of those organisms that I just showed you on the PowerPoint are all going to be example of eukaryotic cells. Most of the creatures that we're going to meet in this course are going to be eukaryotic. And what we're going to see, what makes their cells different from other kinds of cells, is that they're going to have all this compartmentalization, all these specialized structures. And they're going to have what we call membrane-bound organelles. And if you've never seen this term organelle before, you should think organ. So you should start thinking of, your own human organs like your stomach and your liver and your pancreas. An organelle is really just a tiny organ. And these are just these specialized compartments. Remember, if I'm ever talking too fast or going too fast, you can always pause me and slow me down. So eukaryotic cells are what we're going to see in plants, which is a lot of what we're going to see in this course, but we're going to see more than that. We're also going to see them in fungi, just in things like mushrooms and molds. They're also what we're going to see in protists, which are organisms like algae and slime molds. And they're also what we see in ourselves, in animals. So the first model that I want to draw for you here is going to be a model really of this basic plant cell. And so we're going to make this drawing and this diagram of a basic plant cell. And we're going to add in structures and talk really about how it's put together. And this is really going to be our model for the cells we're going to find in all eukaryotes. All right, let's start on the outside, shall we? I'm going to start just by drawing a box. What's on the outside of our plant cells is going to be the cell wall. And the cell wall is going to be this relatively rigid structure made out of cellulose. And there's some other things besides cellulose there chemically, but cellulose is the main thing. We're going to be talking more about cell walls and cellulose in the coming weeks. But the cell wall, it's going to form the outside, right? And it's going to have some really important functions. This is a rigid structure. I'm going to abbreviate the word functions here, just FXNS. I'll try and always explain what my abbreviations mean, but if I ever don't, please ask me. So the cell wall is the outside, it's relatively rigid, and one of the really important functions that it does is really in structural integrity. In general, plants are going to be less squishy than animals because they have these cell walls on the outside of their cells. Now, it's also going to be important in defense. Plant cell walls can signal the rest of the inside of the cell when, say, a pathogen might be attacking, and it might allow the cell to defend itself against something that might injure it. Cell walls, since they're outside, are also going to be really important in what can enter or exit a cell, right? So also involved with what can get absorbed by a cell or what can, and also what can leave that cell. So what can get really excreted by the cell. So that's the outside. Let's move one layer. So I made my cell wall in black here. My next layer, I'm just going to do in light blue. Today, my colors don't mean much. Sometimes they will. Sometimes I'm going to use a specific color because it indicates something. If I do in those cases, I'll be like, grab your red pencil, grab, your green pencil because it's important. In other cases, my colors will just kind of be arbitrary just to kind of help us tell things apart. So just inside our cell wall is our plasma membrane. And that plasma membrane is going to contain the cytoplasm. And the cytoplasm is really going to be the liquid to gel-like matrix within the cell. So that's going to be that liquid matrix within. I'm just going to abbreviate within, just like that. It's going to be that liquid matrix within the cell, and it's going to contain the organelles. So the cytoplasm is going to be everything that's inside of this little blue line. And it's inside of there, we're going to find all these specialized compartments or organelles. Well, in many cells, the organelle that's going to be most obvious is going to be something called the nucleus. And I'm just going to make it, oh, I'll use purple for no particular reason. So the nucleus is kind of circular. And if we've got a nice microscope and we can see inside of it, we can often see some granular stuff inside of it. All of this here is going to be that nucleus. You've probably heard of this organelle before because it's so important. It's what's going to contain the DNA. The DNA, remember, is that genetic information that's inside of cells. It contains all the instructions on how to build the cell and how to express that DNA, right? Which could be through the expression of what organelles get made. If we think about something multicellular like ourselves, it could contain, the ingredients that would eventually get extracted from your body to form things like your hair. So most of the time when we look inside cells and we can see inside the nucleus, we're going to see this little barrier on the outside. That's that nuclear envelope. Right, that's just that nuclear envelope. That's just forming the outside. That's what's isolating the DNA from the cytoplasm. That kind of messy, stringy stuff that I drew inside, that's our DNA. And most of the time, it's going to be in the form of what we call chromatin. So this is just this messy, unwound DNA. It's not condensed into what we call chromosomes, which you might remember from some other courses you've taken in your life. And then the last thing I want to label in that nucleus are those little dark structures. Each one of those is what we call a nucleolus. Now, I know this term looks very similar to nucleus. It literally means little nucleus because it looks like a little tiny circular structure within the nucleus. And this is just going to be some DNA and some proteins, and this is going to be a site within the nucleus where ribosomes get made. Now we're going to be using light microscopes in this course, and they're going to magnify, about 400 times on most of the ones that we're going to use. We're not going to be able to see things like ribosomes. We would need a much more powerful microscope to see ribosomes. So we're going to focus largely in this course on the organelles that we can see with the microscopes, with the tools that we have available to us in lab. Now, one final thing that I want to add to this is I want to add one final organelle. And let's see. I guess I'll just use my pink. This is going to be a big old structure taking up most of the volume of this cell. All right, this big old structure taking up most of the volume of the cell is going to be the vacuole. And so everything that I have there in pink is going to be that vacuole. And the vacuole is going to be a liquid-filled cavity implant. And it's going to be filled with liquid. As far as its functions, well, it functions in storage. Some materials can get stored inside that vacuole. It also functions really in cell rigidity. And you know this if you've ever grown plants, right? On a really hot day, how does the lettuce in your garden look? You know, if you haven't watered for a while, it probably wilts, right? It wilts because the vacuoles inside the cells inside your lettuce are no longer filled with water, right? They're going to lose that water and you're not getting that pressure pushing out on the cell walls. And so it gets limp. But if you water your lettuce, right, that vacuole can fill back up with water. And we can get that, you know, rigidity, that strength in your lettuce plants again.